Thank you, Dr. Lawler, for the nice introduction. Hello and good morning, everybody. I'm going to talk about heritable impacts of germ cell exposure to toxicants because I believe that this is an area that very much needs additional focus in both research and regulation. Oops. Help, Spencer, help. Hold on. Help. So it's not just me.
Just put that on the screen.
Oh, okay. Sorry. All right. So this is sort of a broad conceptualization of how at least I think about toxicology today. Toxicology is very much focused on the adult phase of the life cycle and to a certain degree, childhood phases as well. When we go backwards in time to the fetal and embryonic period, there is, of course, much emphasis on teratology. We're interested in gross anatomical defects, and there's a rising interest in DOHAD, developmental origins of health and disease, and things that may manifest later in life because of early life exposures. But when it comes to the preconception area, when it comes to our gametes, which are a major part of our life cycle, they can last decades, right, before conception, those tend to get the short end of the stick. The post-birth gametes are mostly ignored, but very much ignored are those very early gametes, the primordial germ cells through birth. So in that period, the early germ cells, there are many layers of embryonic germline and fetal germline vulnerability. I'm not going to go over this graph, but basically it shows that the primordial germ cell inside the fetus is rapidly demethylated, and then it's remethylated in a sex-specific manner, and it undergoes or begins to undergo genomic imprinting that persists in the female later in life. There's also a great deal of chromatin reorganization, histone modifications, and repatterning of transcription factors. There are other things that go on, such as mitochondrial effects and X reactivation, which I don't understand. And to the extent that things can be carried through generations, there are also maintenance of methylation in some transposable elements. So, but do exposures during these germline periods, and I'm again particularly, but not solely interested in the very early germline, do they matter for offspring health? And that's a question that I think is unresolved today in science. But I'm going to go through just 3 exposures. I could have, chosen chemotherapy as well or HDAC inhibitors, but these are just 3: synthetic steroid hormone drugs, tobacco smoke, and general anesthesia. So let's start with synthetic steroid hormones. And that's the reason I got into this field. Believe me, I don't have a scientific background, as you heard. But about 9-ish years ago, I discovered that I had been exposed in utero to very heavy doses of synthetic steroid hormone drugs that were sometimes used in some elite practices, fertility practices, for the ostensible prevention of miscarriage. Now, they didn't really work, but that didn't stop practitioners from administering these drugs to their pregnant patients. My mom had 2 miscarriages prior to conceiving me, so the doctor put her on this intensive protocol. Now, discovering this when I was 45 years old, under most circumstances would not have mattered. I'm normal, I'm kind of normal-ish, and I had these exposures. But here's the thing, I have 3 children, and 2 of them have idiopathic nonverbal autism. And as you can tell, my son and daughter are both very good-looking. They're incredibly healthy. They have nothing physically wrong with them. I had no risk factors in my pregnancy. There's no autism at my family tree or my husband's family tree, nothing. And so you can imagine it came as quite a shock to have 2 kids who can't talk, can't read, can't write, can't engage, never have played with a toy, can't engage in conversation, will never be able to care for themselves, have no capacity for abstract thinking. How could this have happened, right? This seems to me a biological emergency of some form. And so when I made this discovery, which is described in some detail in this environmental epigenetics paper, which was published last year, you can look it up. I'm not going to go through the details here. I thought, well, maybe something had happened to my very early developing eggs. Maybe something weird happened that later dysregulated the cortical development of my children. And I discovered, after just talking to some other families with very similar stories, and I'll just share the one here on the left. This is a friend of mine who has had 5 children, 3 of whom have autism, higher functioning forms than my children, but still idiopathic autism. Again, no risk factors at all in this family. It turns out that she was born the same hospital as me, the month before me, in Los Angeles, and had her mother had also had an anti-miscarriage treatment when she was in utero. There's a DES story next door to her, and that's another common story I hear. I have to run through these because I don't have a lot of time. Let's go to tobacco smoke, my second case study. Well, of course, everybody smoked. Really, I think it was almost like 1/3 of women smoked in the mid to late 60s. So it was very common, but this is something I see all the time where you have families where there had been no history of autism whatsoever, and then they have 1 or 2, sometimes 3 children with autism, and there had been very heavy smoking exposure to one of the fetal parents. So here's 2 boys, they're twins, heterozygous twins. They both have very severe forms of autism. Both parents had been exposed to tobacco in utero. And another trend I see is kind of a mix of ADHD and autism in the same family, where one of the parents had been very heavily exposed to tobacco in utero. And usually the girls seem to be more protected. Third example is general anesthesia. That's not just really a fetal exposure, that's also a needle, and through life exposure, a little different. So here are just 3 examples of friends of mine in the Bay Area. The one on the left, she was born in Nicaragua. She has 2 boys with idiopathic autism and no risk factors whatsoever at all. Very normal, healthy family. But she had 2 surgeries as a neonate, and one for a tumor removal and one benign tumor removal and a hernia repair. The friend in the middle, 2 boys with autism, 1 high functioning, 1 very, very severe. She had surgery at 2 days old. My other friend there on the right, she just has 1 child. Her husband had a very, very early life surgery. They have a son with idiopathic severe autism. Now, of course, these are completely anecdotes, and I completely am with you. Like, anecdotes don't say anything. There are lots of people who had general anesthesia as kids, and they don't have kids with autism. That's absolutely true. But I try to use as sort of a mini control group their unexposed siblings. So I ask about their siblings, like, did your brother have anesthesia, and what are his kids like? And in those cases, almost always the siblings' kids are typical. So it's not much, it's just I try to reality check myself from time to time. Here's another general anesthesia exposure that I came across. Not too infrequently, but it's a later in life exposure where there's repeated doses for complicated surgeries. In this case, these are 2 friends of mine, and their husbands both had suffered gunshot wounds in their youths, 1 at 9 years old, 1 at 16 years old, and both of these fathers had to undergo multiple complicated surgeries to reconstruct the damage. In both of these cases, they have sons with extremely severe forms of autism. And just because those of us who are in the autism world see this all the time, but those of you in the research world don't get to see this all the time. I do want to share, I think I just pressed this, just 10 seconds of my friend's son, Pablito. There goes the sound. No sound.
Ah!
I don't need to stay calm.
Ah!
Sit all the way.
Ah!
And that's just my friend trying to give him breakfast one morning. And this is what he's like. I mean, he's obviously nonverbal, obviously severely cognitively impaired, obviously self-injurious to a very high degree. He probably has suffered some traumatic brain injury in addition to the autism because of his self-injury. But in any event, I thought it was amazing that in both cases the fathers had these very unusual surgeries and these very, very unusual and severe outcomes. There are also typical siblings. So to the extent there may have been damage to the spermatogonial stem cells, it probably didn't extend to all those cells, is my guess. All right, so that's my anecdotes, but what about empirical evidence? Well, let's start with the synthetic steroid hormones. The best studied of all agents in terms of intergenerational effects of exposures is the drug diethylstilbestrol, or DES. There's been studies in both humans and mammals. Very rare to have human studies on these types of exposures. And in the human studies, we've seen in the grandsons increased risk for genital tract defects, and in females, There's signs of menstrual irregularities, possible infertility, ovarian cancer, increased risk for preterm birth of their own children, and possibly ectopic pregnancy as well. And then just last year, published in JAMA Pediatrics, was a study from the Nurses' Health Study. This was a study out of Harvard, finding significantly elevated odds for ADHD in the F2s. The F2 means basically the grandchildren of the women who were given diethylstilbestrol. And that was the first time anybody had even thought to ask about neurodevelopmental outcomes in DES grandchildren. Also, there's been pretty much no attention to sociosexual outcomes, and what I see, again, anecdotally, is what seem to be in significant changes in sociosexual outcomes, gender identity, for example, outcomes in the F2s of the DES grandmothers. Remember, these are the directly exposed germ cell. Just to make it clear, I don't do transgenerational inheritance. I only do direct germ cell work. So we're talking about germ cells that were directly exposed in utero. It's basically a toxicology issue and not like a transgenerational issue. So in mammals, there have been more findings findings of cancers and urogenital abnormalities. And again, this is something we had seen in humans. For other synthetic hormones, it's basically impossible to do human work because we don't have the cohorts with the good data. We just don't. Believe me, I've looked all over for them. It's very hard. So the work in that field has been done in rats and mice and some other mammals. With regard to synthetic estrogens, there have been a couple of papers that have found increased risk for cancer, ethinyl estradiol, which, as you know, is in birth control pills, increased risk of breast cancer in the F2 via the F1 female germline, that was in rats. And then Pat Hunt's group in Washington found that multiple generations of this exposure exacerbated the effects, increasing incidence of male reproductive tract abnormalities. Corticosteroids are, of course, an incredibly common drug. I was exposed in utero to the entire first trimester of prednisolone. So they thought it would reduce inflammation, which probably didn't even exist in the first place, but whatever. So corticosteroids are many kinds, and we do find a lot of HPA, I think, related abnormalities, altered cortisol response to stress, altered expression of genes in the prefrontal cortex, and modified brain function and behavior. And you'll notice these studies are a fairly recent vintage. We're just kind of coming up with we're examining these effects just in the past decade, really. Dexamethasone effects on F2's HPA activity. And here was one that didn't look necessarily at neurobehavioral outcomes, but reduced birth weight, differential glucose intolerance, and elevated hepatic activity. And then in mice, again, altered hypothalamic gene expression. That was a very recent paper, I think. Yeah, that was last year. And then, of course, you guys, because you're the NIEHS and also the EPA, you're more familiar probably with the environmental, endocrine disruptor paradigm, for which there's been quite a bit of research. And I'm just pulling out two examples. One's from Andrea Gore's lab at University of Texas at Austin, and she has exposed gestating rats to mixtures here of inclozaline and PCBs, and has found some physiological and social sexual behavioral phenotypes later in life, especially in males. And Emily Rissman's lab, Dr. Rissman is actually here today, here in North Carolina. They've done a couple of studies looking at the generational effects of BPA, and they do find differences in gene expression and also in behaviors here, atypical reactions to novel social partners. Okay, now we're going to go to the second case study, which is tobacco. And there have been a handful of studies in humans looking at the generational effects of tobacco. We just, in fact, held a workshop on that at the Environmental Mutagenesis and Genomics Society last week. We're seeing an elevated risk for autism, and mostly autism-related traits, and now an unpublished study, ADHD, when the grandmother had smoked. And there have been a couple of studies, mostly out of Europe, although there was also one in California, that has found that the F2s had an increased risk for allergy and asthma when the grandmother had smoked. And of course, Dr. DiMarini, who's here from the EPA, has demonstrated that tobacco smoke is a likely human germ cell mutagen, but we aren't. The science isn't quite there to really declare it as such, although it is definitely a rodent germ cell mutagen. And that Health Canada did a fascinating a fascinating perspective. What do you call it? An article 2 years ago, modeling what these de novo mutations in male germ cells might mean for the population-wide intellectual health, and they thought that most likely paternal smoking is having a widespread population, but it hasn't really been detected or researched yet. Now in mammals, we see very similar results. We see the F2s having higher risks for asthma. That's a study out of UCLA in 2012. Another study found these lung abnormalities also when it was just a nicotine exposure. There are studies out of University of Florida State down there in Tallahassee, just on nicotine. This was not whole tobacco, finding that the F2s exhibited the ADHD-like behaviors. So finally, the volatile anesthetic gases. Now, general anesthesia, as you probably know, comes in many, many different forms. I'm limiting this talk to the anesthetic gases, which were introduced in the late 1950s and continue to be used today, although the gases themselves have slightly changed over time. It used to be mostly halothane. Today it's mostly sevoflurane, but other compounds are also used. These are small lipophilic molecules that passively diffuse through cells and enter germ cells and also can enter the nucleus. They work on many receptors. They suppress excitatory paths and augment inhibitory signals. That's how they function. They're very prevalent exposure. Up to 75,000 pregnant women are exposed every year. 6 million children, 1.5 million of whom are infants, are exposed every year. And there are about 48 million surgical inpatient procedures per year. And I just want to pause and say that this is one of the most toxic exposures humans will ever encounter in their whole lives. I mean, we take it for granted that, of course you need general anesthesia. Well, it basically is a lethal exposure. The only reason we survive it is because we're intubated, right? And we're closely monitored. But it basically induces a brain coma. And I'm very grateful for general anesthesia. I have nothing against it, and I'm not angry about it. I just think that we have to keep that in mind. So we do know that general anesthesia exposure does impair germ cell quality, so the integrity of the germ cells. So we've seen negative effects on testicular morphology. These were in animal studies, sperm concentration, motility, spermatogenesis, and this seems to be related to disruption of normal reproductive hormones. Somehow, and I don't really understand how GA does that. It's also seen to be genotoxic, and these have been in human studies, causing strand breaks in blood and increased sister chromatid exchange and micronuclei. I don't think it's known at all as a mutagen, but these were low dose. I want to say all of these studies, strangely, have been on low dose, and I don't know why, but there you have it. And of course, oocytes are routinely understudied relative to sperm, but what little I could find did find a reduction in intact oocytes in an animal model and ovarian injury, and those also seem to be related to alterations in hormone levels. Now, there's been quite a bit of research on the somatic effects of GA, not so much on the germ cell, but the neural effects of GA. When administered early in life, it's known to be neurotoxic, and in fact, 2 years ago, the FDA issued a warning that if possible, a surgery should be delayed till after 3 years old because they were seeing some persistent impacts on neurological and learning function in the exposed children. And mechanisms that they can detect include and aren't limited to neuronal apoptosis, impairments in synaptogenesis, impairments in neuronal migration, including ectopic neurons. So, when the cortex is developing, the neurons migrate from, you guys know what it is better than I do, and then they go up, but they get stuck. and disrupted GABAergic system. And in terms of sort of a molecular effect, it seemed to be a powerful modulator of chromatin remodeling that changes key transcription factors, histone modifications, derails the transcription of the neurodevelopment genes, and has effects on cytoskeleton and synapse formation, for example. Now, what I thought was so interesting when I read the literature about the neural toxicity of general anesthesia was how well it mapped onto what we're seeing in studies of post-mortem autism brains. So these are some of the findings, obviously not all of the findings from post-mortem autism brains, but we find failure of cortical neural migration, failure of cytoskeleton, synaptogenesis, neuron loss, and disruption of the GABAergic system. So of course I'm thinking, well, could this be an artifact of dysregulated gene expression? Not necessarily resulting from a direct exposure, but resulting from a germ cell exposure. Now, no human studies have ever been conducted on the heritable effects of general anesthesia, not one, which I think is by itself a monumental gap in the research. But there have been a handful of mammal studies. These go back to 1981, the first paper published on it out of NYU. And these were small little studies. They weren't molecular at all. They were just looking at behaviors. But they found that halothane-exposed gestating mice had offspring that had exhibited impaired learning function. And at that time, the researchers speculated that the anesthetic agent may have caused a genetic aberration in the exposed mother's fetal eggs. Okay? Then the same lab did another follow-up study looking at a later in life exposure, which was a preconception sire, and they found that the progeny of that sire exhibited also impaired learning function. And again, the lab said it seems likely that spermatogenic changes caused by enflurane are associated with genetic alterations in the germline that affected the pup's brain development. Now, after that, and I consider that to be kind of scary and alarming findings from this lab, there was absolutely nothing in the literature. It's a vast wasteland. Nobody even thought to look at this phenomenon again for whatever reason. And I think it probably had to do with the strength of the genetic paradigm, that this sort of non-genetic inheritance effect probably just was outside of dogma. But anyway, 34 years passed, and last year we finally had another paper out of University of Florida. And this lab, Anatoly Martinyuk, found that sevoflurane neonate rats, which is sort of comparable to very late stage human gestation, their offspring had neurodevelopmental learning and behavioral impairments, and he was able to link it to an epigenetic modification of the gene that he looked at, which was KCC2, which has to do with the function of the GABAergic system. And then he published just this year a follow-up study looking at a later in life exposure, which was again a sire. These were both sires and females. And again, their offspring exhibited behavioral abnormalities, but it was with both of these, the effects were seen in the male offspring and not in the female offspring, which is similar to what we've seen in some of the endocrine disrupting compound studies. And again, it was related to abnormal methylation in both the sperm and in the ovary. Now, Dr. Martinyuk at the Environmental Mutagenesis and Genomics Society meeting last week gave a presentation about his work. He also presented a little bit of unpublished work, but he had looked just at that KCC gene, and here he showed results when he looked at methylation across the genome. And what he says is, I just looked at one gene, but what he thinks he sees, okay, I'll be really fast, is much broader effects across the genome. All right, I have 5 minutes left, so I'll go fast. So, should we test for heritable impacts of germline toxicant exposures? Yes. So I'm here to say yes, we emphatically must do this. And if you want to learn more, I wrote a review or a commentary that was this year in Environmental and Molecular Mutagenesis, speaking specifically about fetal germline. But the 2 points that this commentary raises is, first of all, you have to understand that pregnant women were so heavily medicated and smoked so heavily after the war. So we had decades of very, very heavy fetal exposures. And that we should have a rear view mirror approach and think back to what heritable effects did those exposures have? So we need a rear view mirror approach. We also have to look ahead and think about the people who are experiencing these exposures today. Now, autism. I just have to say this, that there's so much hype in the media about how the autism increase is related to greater awareness, diagnostic substitution, broadening of criteria, blah, blah, blah. I'm here to say emphatically that is not true. That may be partly responsible for this increase, but we have, without any shadow of a doubt, experienced an incredibly dramatic and very serious increase in autism across the country. This is just California data. I'm from California. Where you see the prevalence. This is a Department of Public Health data. The prevalence. This is not caseload. From births in 1990 to 2012, and you can see a steep increase. This is comparing apples to apples. These are just the cases that enter into the developmental disability system. These don't include the high-functioning cases. And in fact, we restricted, in about 2003, the state restricted the entry requirements. We made it harder to get in, not easier to get into the system. But now, fully 2% of all boys born in California end up as DDS autism cases. There's no precedent for that. You can't possibly find anything like that in prior generations. So it's, to me, obvious that this is real and it's happening. And the CDC numbers are actually very similar, but they're bigger because they include more people on the spectrum, 1 in 59. Now, this is a highly heritable disorder, but it's not proven to be highly genetic. And when we have seen genetic causes, they're mostly de novo mutations. So there's a great deal of missing heritability. So I also want to point out that this hypothesis happens to be consistent with a lot of autism epidemiology and phenomena. There are temporal associations. The start of the autism increase was observed to have begun with births in the early 1980s, and that kind of maps onto a heritable effect that would have been in the post-born decades. Strong heritability. Again, we're not finding the heritability. The male 4-to-1, male to female 4-to-1 ratio. That could be explained by these heritable effects. Autism's heterogeneity. If it's not a pure genetic factor and it's maybe a mix of genetics and epigenetics, you're likely to have a whole bunch of different outcomes. Parental age effects, maybe those are more related to the cumulative effects of exposure over time. Some regional, socioeconomic, and ethnic disparities, although those seem to be reducing now. And also the fact that autism clearly arises in early brain development and is clearly related to the birth of those cortical neurons. But today, really, nobody really thinks about our germ cells, and really nobody thinks about our early germ cells, for sure. I'll just run through this. The FDA, they just don't do it. I've petitioned them twice to do it. I have friends who work in the FDA research arm, and they just say, we just don't have the bandwidth to do it, basically. The FDA Center for Tobacco Products, they really don't think about the heritable effects. They're very concerned, and they should be, with the somatic impacts, heart, lung, stroke, et cetera, very important endpoints, but germ cells not on their radar. EPA, a little bit. They do, Ames assay type mutagenesis work, but not looking at maybe other mechanisms that could be involved. The NICHD does some basic germ cell science, but they don't do anything related to exposures. The NIMH, they really focus on genetics, but really haven't looked at the context for possible genetic change. Now, the NIEHS is actually a bright spot because this institute does quite a bit of looking at germ cells and heritable effects. You did a scoping review on transgenerational effects. I submitted a nomination that you guys should really look at intergenerational effects before you even think about transgenerational effects. Transgenerational effects, there's no direct exposure to the germ cell. Probably more important to look at the generation where there is a direct exposure. The CDC, which does a lot of autism work, doesn't really think about past exposures. But I can dream, and I'm dreaming about the Manhattan Project for germ cell exposures. What would that Manhattan Project look like? Well, it would look at various exposures that really raise red flags for possible genetic and epigenetic toxicity. I've listed some of them here. It would invest heavily in epidemiology in the Nordic countries because they're the people who have registers and where you can do some generational work. Sorry, US, very hard to do the work here. And there would be intramural work in the lab and also an extramural grant program devoted to this. And then again, this OHAT nomination on a scoping review, just to kind of compile what we know today. What do we know today? I think that would be a very important exercise. Final thoughts. There are not 2, but there are 3 routes to pathogenesis. Genetic, inherited, where we've put a lot of money, environmental, somatic, and I think there's this 3rd route, which has been pretty much ignored, germline exposure. But I do think we should be especially protective of that particular part of our life cycle, because that is most likely our weakest link. Thank you very much.